Okay, DNA take 2. Messed up the first one. DNA, deoxyribonucleic acid. What we all have, and in fact, all quote-unquote living organisms have DNA, and they pass it to their new cells, and they pass it to their offspring. So, DNA, we're going to look at its structure and its organization, and we are going to look at DNA replication. Structurally, DNA exists as a double helix. You can see that up here, okay? It's, we'll look at more detail in that double helix arrangement in a little bit. But, in fact, most of the time in the cells, it's all unwound. It's in not just the cells, but in the nuclei of the eukaryotic cells. So it's all unwound. In fact, watch that video and get the idea of just how much DNA there is in these nuclei of the cells. Okay? So when it comes time for the cells to divide, we have this process specifically called mitosis. And for mitosis to occur, the double helix, all that double helix structure in the cells has to be condensed. And basically what it does is it wraps around these proteins. In eukaryotes, they're histone proteins, wraps even more until you see the familiar representation of chromosomes. Okay, so we've got DNA here, we've got chromosomes here. And the thing is, this is much more complex than what you're led to believe. For one, there are 2 sister chromatids present. There's one here along the top and one here along the bottom. Those are the products of replication. So we'll come back to that. Okay? So the DNA molecule itself has multiple genes on it. So the genes are the parts, and there can be many of these along that whole linear strand of DNA for a given chromosome. There are genes, and genes are those parts of the DNA that hold the recipe for making proteins. Each gene makes a particular protein. That is what most of them do. Now, there are also genes that make ribosomal RNA and transfer RNA. Someone asked about, hey, what about these RNAs? And we're going to learn more about messenger RNA. That's directly to make the proteins. Transfer RNA in translation, and ribosomal RNA that makes up, well, the ribosome. So we'll learn about those some more. We're just not quite there yet. So, and RNA stands for ribonucleic acid instead of deoxyribonucleic acid. I mentioned gene expression. Gene expression is defined as the process whereby genes direct the synthesis of proteins. And you need to know that, okay? You also need to know, and again, next week we're going to look at this some more. What happens is DNA is transcribed into RNA. Now, if it's for transfer RNA or ribosomal RNA, this part doesn't happen, okay? Because that's all that needs to happen to get those particular types of RNA. But what we're really going to look at, and what we're really interested in, is the whole gene expression, where DNA is transcribed to messenger RNA, and that is translated to proteins. Okay, so you can see DNA transcribed to RNA, and RNA translated into proteins. And this is specifically messenger RNA. So just to get a feel for these and to get an appreciation for all our DNA that's wrapped up with proteins forming our chromosomes, I wanted to show you these and then talk about karyotypes just a little bit. This is a karyotype of a human female. There are 23 pairs of chromosomes. You can count them if you don't believe me. 2 1s, 2 2s, 2 3s, all the way down to 2 22s, and then 2 X chromosomes. Okay, so this could be my karyotype, for example. It's not, but it could be. These are actual human chromosomes. These are the chromosomes of a female. Okay, when they're most condensed, when they're in kind of that X form. So that this one is one of the X's, and this one is one of the X's, and this one is one of the X's, and this one is one of the X's that we saw. So that this has 2 sister chromatids, this has 2 sister chromatids. So these are made, the karyotypes are made when cells are getting ready to divide, or even starting that process, so that the chromosomes are as condensed as they're going to be. All right, so 22 pairs of autosomes, and I'll get to that later, and then the sex chromosomes. For 23 pairs total, 46 chromosomes. This is a male. So we have the 2 1s, the 2 2s, the 2 3s, down to the 2 22s, and then an X and a itty bitty Y. Okay, you see the Y chromosome? It's kind of small. So you have, these are paired, and you have one from mom and one from dad. One from mom, one from dad. They're not necessarily in that order because they're not pink and blue. You can't tell in a karyotype which chromosome came from mom and which one came from dad. The only way you can tell is with the Y chromosome, because it came from dad to make a male. Okay? But they're paired. And for each pair, like, we don't know which one, but one's from mom, one's from dad here, one from mom, one from dad. The rest of them we can't tell in the pairs which ones are from mom and which are from dad. Now, they don't exist like this in the cell, in the nucleus. These are done by technicians who do karyotypes, okay? Over here, we have a different karyotype. It is a female, okay? The karyotype is of a female. It's not an it, she. There's 2 X chromosomes. But notice in the circle, there are 3 21s. That is one of the primary reasons that karyotypes are done. They are part of an amniocentesis. So basically, the pregnant woman goes in, she has some amniotic fluid taken out, they grow cells in tissue cultures. They can't grow new babies, just cells. They can separate mom from baby cells. So they're looking at the fetus, the cells of the fetus, and then they do the karyotype to determine how many chromosomes the individual has. That's one of the primary uses. And one of the primary uses is to definitively diagnose Down syndrome. So no ifs, ands, or buts, this individual has Down syndrome because there are 3 of chromosome 21. Instead of having 46 chromosomes, this individual, this female, has 47. Another thing that can be noticed from these are what are called deletions or misshapes of these chromosomes as well. They kind of, for us, it's kind of, some of them are kind of weird shaped and all, but these technicians know what they're looking for. So they can tell if there are other issues besides chromosome number. Now, I do have another, we call these trisomies, when there are 3 of each chromosome instead of the paired. And this is a karyotype of a male with Klinefelter syndrome. How do I know? It's not because I am really an expert at reading these, but I can count. So and someone has put them together, and I see there are 2 1s, 2 2s, 2 3s, all the way down to 2 22s, and then look, 2 X's and a Y. So this individual is chromosomally a male, but a male with Klinefelter syndrome because he has an extra X chromosome. So hopefully that clears up some of the information about karyotypes, and maybe you get to see how neat this is when this is done to determine if a fetus has the correct number of chromosomes that are all correctly shaped. And they can't diagnose genetic disorders with this. These are chromosomal abnormalities they're looking for, not genetic disorders. That takes DNA testing. But yeah, it is still incredibly neat. I was discussing this one time with a class of students, and this young man says, "See this scar on my face?" Said, "My mom had an amniocentesis and they poked me with the needle." So his first wound was before he was even born, when he was a fetus, somewhere around 14 to 16 weeks in development. Interesting, huh? We'll come back to chromosomal abnormalities later. So let's talk about DNA structure. The double helical structure of DNA. This. Now that's without the proteins, so we're not looking at the chromosomes. We're looking at the chromatin, okay, the actual DNA. And if you look closer, what we see are the building blocks. This has already been mentioned early in the quarter, which was just a couple of weeks ago, right? So this is a nucleotide, and this is a nucleotide, and this is a nucleotide. Oh, and there's nucleotides over here too. Those are the building blocks. So each nucleotide has in common a phosphate group, a sugar, and if it's DNA, the sugar is deoxyribose, and then a nitrogen base. And you can see there's a G here, there's a C, there's an A, there's a T. Those are the 4 bases. That's the whole alphabet of our genome. It is so darn cool that from those 4 we get all the different proteins that make us so unique. It is just, it's crazy. Anyway, so this is guanine. That's what this nitrogen base is. This one's cytosine. This one's adenine, and this is thymine. Okay? So the information for a protein is in 3 of these that are adjacent. So if this was the beginning of what will result in the codon, the messenger RNA codon, AGT, and we'll look at this next week, is for an ingredient, a particular amino acid. Just like ACG is also another amino acid. And we'll talk about how fantastic and how interesting and cool all this is next week, but not quite yet. We know that Watson and Crick are the ones who received the Nobel for determining the shape of the DNA, but without Rosalind Franklin, they never would have done that. And really, she should have been honored with this discovery as well. Her name should be on it, not just Watson and Crick, but unfortunately, she was too nice and they weren't quite as nice. Okay, so what else? If you think of this as a ladder, okay, let's think of this as a ladder. What we have are two sides of the ladder. And the two sides then, the structure of that would be sugar phosphate bonds. See that? Sugar phosphate bonds. Same thing on the other side, sugar phosphate bonds. Okay? Then the steps would be base pairing between the nitrogen